years here since joining MSU. I guess I'll start by just noting that the naming of this seminar is after Jim Hanover. And so Jim Hanover started in the department in 1966, kind of following on the heels of one of the very first forest geneticists, which was Jonathan Wright. Jonathan Wright wrote the first textbook in forest genetics. And so when Jim Hanover started, he kind of picked up on some of the work that Jonathan Wright was doing. And then by 1974, he proposed to start what is we call now the Michigan Cooperative Tree Improvement Program. And so that's kind of an important aspect of my research. So much of the focus of my research and the direction that I want to take my research really hinges on the Michigan Cooperative Tree Improvement Program. Now, of course, in the 1960s, 1970s, into the 80s, the focus of forest genetics was maybe a little bit different than where it's going these days. We focused a lot on pulp and timber production, identifying traits that are important for growth, to fast growth for economic reasons. And now we've shifted more towards assessing adaptability to climate and some of those types of traits. And so I think it's important to note that although the questions are maybe a little bit different, the need is still there. We want to have durable, sustainable, resilient forests for the future. And so I'm going to spend some time today talking about my research, thinking about how we can implement genetics and genomic approaches to identify conservation needs, to identify areas of maladaptation, traits that are adaptive to the future, so that we have these durable forests. And I was watching Asia's seminar last week. If you had a chance to see that, it was really well done. So I don't really need to introduce this too much, but one of the things that I have benefited from in my appointment at Michigan State University has been a PERM appointment. And so PERM is the Partnership for Ecosystem Research and Management. And really what this does is it facilitates my role as director of the Michigan Cooperative Tree Improvement Program. And I'll speak more about MichigoTIP towards the end of the seminar, but I do want to just state that this has really allowed me to interact with the state DNR, Forest Resource Division in particular, in terms of identifying priorities for the state, the directions that they want to go in terms of tree improvement, and using forest genetics to help develop management interventions and decision support tools. I use a co-production of science approach with them. We meet every week on Wednesdays, talk about what's going on with the forest, and then when we have issues or problems associated with development of forest genetic resources, come up with an idea to implement a project that might be able to address those questions. Okay, we have some technical difficulties maybe. It's fine. So what I'm going to do here is we're going to jump into, I guess, some emblematic examples of the research that I've been conducting using genetics and genomics to address a myriad of issues ranging kind of from climate change issues to forest health, pests and pathogens, and then also thinking about landscape genomic approaches in forestry as well. I wish I had a clapper and some slides there. Okay. Is everything good? We're good? All right. So getting going here, we're all aware that temperatures have been increasing over the last decades. In particular, we think about this year, we're on track for the warmest year on record. But again, this is not news to anybody. We hear the same kind of story every single year now, and this is starting to be quite problematic. And because of this increase in temperature, because of the changes in the environment, the future of forests are really uncertain. There's a lot of variability in terms of the spatial patterns of how forests will respond to climate change. So this is a paper that was published in Science by Bill Andrig in 2022 that was looking at several different modeling approaches to understand the risk that's associated with climate change. And so you've got these three panels here that kind of show similar spatial patterns, but what we notice is that for some of the largest forested regions in the world, we have increased threat to climate change. And so if we just look at panel C right here, for instance, we see the boreal forests are quite red there. That is indicating that there's a large risk for species turnover. So loss of species, change in species richness, The other panels show transition from one ecotype to another. But what we expect by 2070 is that there's quite a bit of change that will happen in terms of the actual composition and configuration of forest environments. That's really concerning because it's 2024 right now, so we really don't have that long to think about the types of interventions that might be needed to help create these durable forests. In 2008, Sally Aiken out of the University of British Columbia and her colleagues proposed what is a pretty famous paper now, looking at some of the alternative responses of forests to changes in their environment. And Sally proposed that there were three kind of scenarios that we could expect from forests. And the first one is adaptation, right? So based on the underlying level of genetic variation that exists within species, they should be able to adapt, right? So if there's higher genetic diversity, there's more possibility for a population to respond to changes in its environment. And then they should be able to persist in situ in this changing environment. There's also migration, right? So as climate niches shift, perhaps species can track that shifting climate through dispersal, seed dispersal, of course, to expand their ranges, but also potentially gene flow through pollen that would allow adaptive alleles to introgress into populations and make them more adapt to the future. And then the idea of phenotypic plasticity. So the level of maybe intraspecific variation that exists within individuals, the change in a phenotype that allows them to respond to the environment without a change in genotype, right? This is classic kind of phenotypic plasticity. And these are not mutually exclusive alternatives for the forest to respond, but probably interacting together to allow them to persist. And of course, if forests are unable to take advantage of one or all of these scenarios, then we would expect to see some level of extirpation within a portion of the range, probably at the trailing edge of the range, but maybe at the northern margins or in isolated populations that are kind of isolated and trapped from being able to disperse or move or respond. So this brings me to kind of thinking about how we can use genetics and genomics to assess populations for maladaptation, to identify where they might be most imperiled and where we can then target interventions that allow us to make good decisions for sustainable forests. Luckily, what we can do is we can assess the inherited components of variation within populations, within species using genome scans. So this is a figure that I forgot to put the reference onto, but this is a figure that I created for a paper in Biological Reviews a few years ago that was led by Sean Hoban out of the Morton Arboretum. And I think it does a good job of kind of simplifying what we mean by looking at the genomes to understand variation within there, right? So when I talk about variation here, I'm just going to use a simple metric. This is an essential biodiversity variable that you can use across organisms and taxa, but it's looking at heterozygosity. So when I say heterozygosity, I'm saying you have two alternate states of a gene. If you're a diploid organism, you have half from your mother, half from your father. So you might have two alternate forms of a gene. So in this figure here, we've got species that run across, and then we've got basically the gene pool that kind of goes down through the columns. And if you look at sample one, we've got a genome sequence. We've got both sides of the gene. So if it's homozygous, the same, you have two exact copies. If it's heterozygous, it's alternate. So as we go across, we tend to take a sequence and identify molecular markers that might be informative for telling us something about the genetic diversity. So the genotype, L1 through L4, gives an example of that. And if we look then across that, we can say heterozygote, homozygote, heterozygote, homozygote, and use that to calculate the actual frequency of heterozygosity. So in the first example, it's 50%, right? So reasonably good heterozygosity, what we might expect from theory. You can look across the other samples, 50%, 25%, 0% heterozygosity, right? So in the last case, we have all homozygosity. There's no sort of level of diversity in which the environment could act upon to increase fitness. That would potentially be the most at-risk individual. We can also look through the population at the gene pool. I guess I should stress that when we talk about evolution, the population is the level, it's the unit in which evolution acts. It's not on individuals. And so we might be curious about what the heterozygosity is at those loci, at those genes across the population. So you can do the same thing. Look down through L1 through L4, and you have 25%, 25%, 50, and 25%, right? I'm going to keep coming back to this idea of genetic diversity, which is why I'm providing this kind of background context here. So when we have populations and we're trying to understand where they may be more or less fit in the future, we might start by just considering these very basic essential biodiversity variables. So the paper that came from, again, led by Sean Hoban, this was a kind of large undertaking through the GeoBon group to develop a set of essential biodiversity variables for genetic composition. Right, what kind of universal variables can we look at across the range of species or cross populations and draw some kind of conclusions from? And so what we came up with were these kind of standard population genetic metrics. I've talked about genetic diversity here already in that first figure that I showed, but looking at heterozygosity or allelic richness would be examples of that. In this figure, I created kind of greenish yellow boxes to show maybe the desired state. And so in this case, we'd like to see higher genetic diversity, right? We'd like to see the genetic differentiation is a little more complicated, but in general, what I would suggest is that we'd like to see lower levels of genetic differentiation. That suggests higher levels of gene flow shared between populations. So they're not isolated, they're not restricted by changes in the environment. The level of inbreeding, we'd like to see lower levels of inbreeding. That generally will result in a higher level of genetic diversity, heterozygosity. And then lastly, this idea of effective population size. Go out in the forest, you see a lot of trees there. The truth is they're not all contributing equally to the number of genes in the gene pool. Some have higher fitness, are more likely to reproduce, and those that are actually contributing to the change in allele frequencies within a gene pool, that constitutes the effective population size. So higher effective population size, low inbreeding, lower genetic differentiation, and then high genetic diversity. So if we just go out and sample a forest, that gives us a good idea about the current state of its gene pool and whether it might be at risk of maladaptation. We can look at these kind of EBVs and use genetics at a variety of scales. I tend to focus on population scales, but as I get into some of the research that I've been doing, you'll see that I look at various aspects of this. So species level, using something like structure analysis to identify different genetic units. What are the optimum number of populations? Getting down in phylogeography, for instance. Populations, this is where I spend a lot of time looking at richness and diversity, looking at the effects of inbreeding and genetic differentiation. And then also at the individual level, which is oftentimes not considered, there's a lot of interspecific variation that exists between individuals. So all that background was just to kind of give you some context for what I want to show you with my own research here. So within the Forest Genetics Lab, we really focus on 4 different kind of research avenues: landscape genomics, dendrogenomics, forest health, so really thinking about disease resistance, and climate adaptation. And the overall goal of the lab, kind of mission statement that I have, is to make this research translatable, usable in terms of interventions for improved forest health, sustainability. Created this little conceptual figure here, because even though the particular research areas might change through time, overall what we wanted to do is create synthesis and translation so that the basic science can be applied towards real world uses. And a lot of times that's through tree improvement breeding approaches. So what I want to do for the rest of this talk is I'm going to start out by giving a few emblematic examples of this type of research: landscape genetics, genomics, dendrogenomics, and then some disease resistance work. I'm going to highlight what I've done recently and then some of the work that's going on in my lab by the grad students. Then I'm going to shift gears and talk about Michigo tip and some of the future work associated with assisted migration and climate adaptation. So I'll start with landscape genomics. This, for those of you who are not familiar with landscape genomics, it's really kind of a branch of population genomics, and it's a framework or an approach for assessing how the configuration and structure of the landscape influences the movement of genes and thus population structure and gene pools, right? And so it allows the identification of breaks within these populations in terms of the genetic similarity and correlates that with environment, landscape, structure. So if we look at this figure right here, we've got 3 different patches embedded within a landscape. This is very kind of landscape ecology, classic theory. And they're all embedded within this matrix, right? And the matrix represents, in some sense, the permeability in the landscape, how well individuals can move through it. In this case, we'd be thinking about pollen or seed from trees. And the different colors might represent the level of genetic differentiation. So you see that there is some variability between those patches. How similar or dissimilar they are might be related to how easily they move across that landscape. And so I've been conducting kind of landscape genomic research since about 2011, really my dissertation work. And I've done this at a variety of scales from site level movement, parentage analysis, all the way up to macro systems and range-wide distributions. So this first paper that I want to highlight is a paper that I worked on and collaborated on. This was published this last year, and it was assessing the relationship between red spruce and black spruce. A lot of it is thinking about how red spruce population genetics have been impacted by historic glacial cycles. And think about, say, the current distribution of species in North America. And so much of this is influenced by kind of transitory ice sheets that are a result of the different glacial cycles. And the last glacial maximum, as the ice sheets receded, this led to the expansion of populations, and red spruce was one of those. And what's interesting about that is that when we think about the time scale that's involved with forests moving in response to these kind of historic climate events, the frontier populations that are leading the advancement may only be a few generations old in a lot of cases because of the long lives of trees and the amount of time it takes for them to move across the environment. Also, as a result of this, they're most likely coming out of these glacial refugia or microrefugia, and you get mixtures of lineages that interact together, and this leads to really highly dynamic gene pools at those leading populations. And then at the trailing edge, you have species that are moving up from the south that are probably better able to compete for habitat because they're pre-adapted to the warmer environments, and so you have this declining population at the southern margin. So red spruce occurs along the Appalachian Mountains, all the way up into Canada. And there's a sympatric zone where red spruce exists with black spruce, kind of at the northeastern portion of that range. And there's a lot of introgression of alleles that are occurring there. And there's been some hypotheses about the origin, the evolutionary history of red spruce. Some folks have proposed that it's a derivative species of black spruce. Others have suggested that it's potentially they evolved from a common ancestor and that they have had different evolutionary histories. So we set out to answer that question. The strategy for this was to sample 8 populations of red spruce along the Appalachian Mountains and up into Canada. Each of those populations had 60 individuals that were sampled, and we used a magnetic fishing approach with beads to extract the genomic DNA out, and then use some highly polymorphic nuclear microsatellite markers. In this case, we had 9 of them to do the genotyping. And from there, when I analyzed the data, I did basically what I had just kind of introduced. I looked at the level of heterozygosity, level of genetic differentiation, effective population sizes, inbreeding, all that kind of stuff. And then I used 2 different Bayesian approaches to assess first, what is the optimal level of populations that exist in the landscape, given its evolutionary history. And then I wanted to test the alternate scenarios from the kind of phylogeography standpoint. What was the most likely pattern of evolutionary history? Did red spruce diverge from black spruce? Did they diverge from common ancestors? What explains that current distribution? So this is the, this map shows the different sites that we sampled. So there were 8 of them from the red spruce populations that range from Tennessee all the way up into Nova Scotia. The shading on this is maybe a little hard to see. The dark gray is black spruce. The red is the red spruce. And then that kind of grayish area is the sympatric zone where the ranges overlap. And then we have 2 populations of pure black spruce that were pretty far away from the red spruce range, kind of as controls. All right, so the results. From the first set of analysis, the structure analysis, this is basically trying to figure out what is the optimal number of populations that truly exist given the multilocus genotypes that we had in this population or in this sample. This is what we call an Ivano plot. It's just estimating the most likely level given the data, the priors in there. And so the higher the delta K variable, the more likely the situation is. And so you can see here pretty clearly that you have a population level of 2. It's saying that the most likely division between all of those samples is at 2. I'm going to show you a plot that illustrates this better, but it makes a lot of sense biologically because we have black spruce and red spruce. So that's not that surprising, right? You hope to see a very strong pattern in that. But there is substructure at K equals 3 and 4. So if we look at a structure plot here of that, and what these plots are, these are like bar plots, but they show the proportion of ancestry for each individual in the data set. And so at K equals 2, where we see that kind of red spruce, black spruce pattern, we have all of the blue is red spruce and the orange is black spruce here. When we look at the southern portion of the range, Tennessee, West Virginia into New York, it's almost all just red spruce. And as you move further north to Maine, Quebec, into Canada, you see more of the ancestry being a component of black spruce, right, in this kind of hybrid zone. And then the black spruce populations in New Labrador and Manitoba are pretty much just black spruce with some introgression of red spruce alleles. So that explains kind of the difference between the species, even though they can hybridize. But if we look at K equals 3, which was the highest level for the substructure, we see that Tennessee has a very distinct cluster compared to the rest. So the furthest south site looks like pure red spruce. And as you move into that kind of West Virginia, New York area, there is still a lot of genes coming from the Tennessee populations. Then you get further into the northern red spruce range and finally into the black spruce. So because of that pattern, I selected that to do the rest of the analysis using an approximate Bayesian computation. And just note here, these boxes at the bottom, so we've got the southern red spruce as kind of a coherent population, northern red spruce, which represents sort of that overlap in that sympatric zone, and then the black spruce. So the models that I tested, I chose 3 different models, and the colors correspond to that structure plot right here. So we've got the southern red spruce, kind of like that Tennessee color, northern red spruce in blue, and then the black spruce. So the first scenario is an ancestral scenario. Just all of those groups originated from a common ancestor. No other explanation for it. A hierarchical scenario, which was that red spruce and black spruce split from a common ancestor, but then we have another event that splits off that northern red spruce from the black spruce. And then an admixture scenario where you've got red and black spruce diverging from a common ancestor, but then you have recontact and hybridization at some point later in the past. And so those models were then used in the approximate Bayesian analysis. I was able to take the relative posterior probabilities, and I computed a number of sample statistics, the number of alleles, mean genetic diversity, FST, which is a measure of genetic differentiation. And I simulated that 1 million times. And from that, I took 1% of the posterior probabilities that were closest to the observed data, and I used a logistic regression to pick the most likely scenario. So in this case, the admixture scenario was by far the most probable. So from that model, I then estimated the parameters in terms of the kind of evolutionary history. So if we take the admixture scenario and I estimate the parameters, importantly, what this tells you is the number of generations at which the data is suggesting something occurred right here. So if we look at time 1 here, 343 generations, it's suggesting here that black and red spruce diverged from a common ancestor around 124,000 years ago, and that following the last glacial maximum at the beginning of the Holocene, you've got recontact. And the dates work pretty well for that, almost 10,000 years ago. The other thing that I was able to estimate was the patterns of gene flow between those populations. And so RA is gene flow from red spruce into the northern red spruce, southern red spruce into northern red spruce, and 1 minus RA is the reciprocal of that. And so what we see is that 3/4 of the gene flow is coming from red spruce into that northern red spruce population, and that 1/3 of that is coming from black spruce. So this was pretty interesting work because there hadn't been a lot of evidence talking about the evolutionary history of these two spruce populations. And I'm pretty excited about the results there. But we think about Tennessee and West Virginia populations being maybe remnants of pure red spruce glacial refugia. This matches with some fossil evidence by Jackson et al. In 2000. So the other thing to consider is the kind of vegetation dynamics of all of this, the successional stages, the difference between red spruce, which is a late successional species, and black spruce, which tends to be an early successional species, that impacts their migration rates. And so if the Tennessee populations were a refugia for red spruce, then they moved towards New England, cooler, wetter environments following the glacial retreat, and the black spruce migrated out of the Mississippi alluvial valley at a kind of faster rate, where they recontacted after about 10,000 years ago, and starts to generate that kind of sympatric hybrid zone where all those alleles are coming in. The other thing that this paper did, and I don't have all the tables in there, is but we were able to estimate the levels of genetic diversity and gene flow between the red spruce populations. And you clearly see that the southern red spruce populations that are isolated on the mountaintops or in these cool, moist areas have a much lower level of genetic diversity than they do in the sympatric zone, and even compared to red spruce. They do have a number of private alleles. They are coherent in terms of their genetic architecture. That's an example of some of this type of research. Now, what that paper didn't really do, we looked at species level differentiation, so the difference between red and black spruce. We looked at population level analysis, thinking about diversity, effective population sizes. Those are all in that paper as well. But it doesn't really discuss individuals or patterns of, say, intraspecific variation, which I think are just ignored in a lot of cases, and we need to really start exploring some of that. So this brings me to this idea of dendrogenomics. So dendrogenomics, this is an area that I actually think that I'm pretty well positioned to make MSU kind of leader in this field. This is kind of an emerging genomic field. And the reason that I think this, so this really stems from a paper that was essentially a tack-on paper at the end of my dissertation. Had not set out to do this type of study. And I was working on landscape genomics of mountain hemlock in Alaska. I had a lot of different sites, and we had done genomic sequencing. So we had thousands of single nucleotide polymorphisms, these kind of molecular markers that we can use in genomics. But one of the sites that I had was in the Palmer Creek drainage on the Kenai Peninsula in Alaska. And what I had done there was I sampled exhaustively every single mountain hemlock on a mountain slope, right, from alpine tree line down. So I had all of these transects. I had cored all of the trees, or I had taken cross sections. So I had all of the kind of tree ring data, and I had done all of that for a parentage analysis. But at the very end, after I'd actually finished, I thought, well, I've been reading these papers about heterosis and homeostasis, thinking about how trees grow in stressful environments. And it seemed pretty clear that like one hypothesis would be that if you had higher genetic diversity, you might grow better in stressful environments. So I did this real quick study on a single transect. And what we see here, I took average tree ring width, variances in tree ring width, and I correlated those with heterozygosity, average individual heterozygosity, not population level. And you can see that there's a slight positive trend in the data as heterozygosity goes up, average tree ring width increases. However, it's not significant here, right? But what was significant was that variance in tree ring width was correlated with average tree ring width. My conclusion in that paper was just that all that really suggested was that trees grow poorly in poor environments. And it didn't have anything to do with the genetic diversity. But I continued to think about that. And I don't know, it's just that the data set itself was too limited. But I think that there's more to it. And just using average tree ring widths as kind of a dendrophenotype isn't maybe the most appropriate way to assess high resolution patterns of growth and its relationship with genetic diversity. So after that paper, I don't think that I had called it dendrogenomics in that paper, but I wrote a book chapter for the spring or population genomics text that came out titled Advancing Biogeography Through Population Genomics. And in there I said, this is kind of an emerging field called dendrogenomics. Since that time, there's been several different papers that have come out trying to analyze dendrophenotypes and relate that back to kind of genomic diversity. And so this whole idea of dendrogenomics is that most of us are probably familiar with dendrochronology. Take a core out of a tree, calculate the basal area increment or the tree ring widths, correlate that with climate, say something about the reconstruction of climate from the growth rings, which is a very useful approach. And we've talked about population genomics here. So we've heterozygosity, we've got all these different essential biodiversity variables that we might be able to link to patterns of growth. And then quantitative genetics, this is kind of what we do in tree improvement. We look at a phenotype, which might be the growth rings, and we estimate the proportion of heritability that is explained there, given the environment. And then all of that together, we can come up with dendrogenomics. This figure, where's Raju at? Raju made this figure, and it helps to explain kind of the ways that we go through this. So a dendrophenotype, I'd like to think about this being a little bit more than just those tree rings. We can take a tree ring, we can scan a high resolution image of that, or we can take a microtome and take a thin section of that and mount it on a microscope slide. But what you can really do is develop phenotypes that are based on wood anatomical traits. So if you're curious about, say, patterns of growth within a year, so not just the ring width, but like the density of the cells that might exist in that growing season. You might be interested in hydraulics. So the trade-off between cell wall thickness and lumen diameter, or some of these types of physiology, you can calculate phenotypes from that and then link that to the underlying genomics that help regulate it to say more about how individual trees are growing at various points within their range. So if you think about the role of range margins, both northern range margins and southern range margins, and how climate changes in those locations, they're ideal systems for exploring the response of trees to that change. So we got my lab, we got Raju Beast over here, is my PhD student. He is leading the charge on this project looking at white spruce dendrogenomics. This is a collaboration with several other researchers out of Alaska and Massachusetts. Dr. Patty Sullivan is at University of Alaska, Anchorage. Roman Dial is at Alaska Pacific University. And then Dr. Becky Hewitt is Amherst College. And they're working on various aspects of this question, looking at soil, looking at climate, looking at the patterns of sea ice and the change in snow depth that's associated with the change in open water as a result of loss of sea ice. What we had is a nice precipitation and temperature transect that runs along the Brooks Range in Alaska. Patty had talked to me several years ago about noticing that white spruce was beginning to jump over the Brooks Range. So we're seeing a shift in latitudinal tree line, which is important. So where those trees are coming from, how they're growing in response to the changes in climate. This is what Raju is currently working on. And so we have 1,300 samples from these 20 plus sites. They range from high elevation to low elevation sites, and they are classified as responders and non-responders in terms of their growth patterns related to snowpack and soil and some other characteristics. We've extracted Raju has extracted all the DNA out of this. He sent that off. It's being optimized right now for a reduced representation sequencing project. We have all of the cores, developing the phenotypes from that, and I think he's going to have a great dissertation from that. So I'm excited about this project. I have a collaborator in Canada that I work with frequently. He's the one who I worked with on the red spruce project. He's also sampled 300 samples across the middle of the range in white spruce. So those are being sent over to be included in the analysis as well. And then the plan is to go out and sample the southern range here in the Lake States and look how they're growing in the south as well. Okay, so let me switch gears real quick and go to forest health. We think about the risks that are associated with climate change here as well, but pests and pathogens are also a threat to our forest. This is the National Insect and Disease Forest Risk Assessment. It's a little bit outdated at this point, but I think what you see is that there's pretty significant risk in the Western United States, but also when we look at Michigan, there's quite a few threats that exist here too. And there's other maps that can illustrate this pretty well, but because of the population size of Michigan, the connectivity of the Great Lakes, the movement of goods and services, we have a lot of pests and pathogens that exist around here. We also have pretty high species richness in terms of our species, and so we just have a lot of opportunity for pests and pathogens. I was in Vermont last week at the Northern Forest Genetics Association, and we went and visited a bunch of different trials. I took some photos just thinking about the threats that exist kind of in this northern landscape. Beech bark disease, talk about here in a second, Mitchell's work on beech bark disease. We've got American chestnut blight. This has been a problem for a long time, right? And so we're very interested, the state of Michigan is interested in trying to start thinking about reintroduction of American chestnut. What kind of resistance do we have in there? Things like butternut canker disease, Dutch elm disease, hemlock woolly adelgid, beech leaf disease. The list goes on, right? This is pretty significant problems. So from a genetics perspective, how do we deal with genetic resistance to these pests and pathogens? It's another area that I've been working on for quite a while, mostly within the white pine blister rust pathosystem on a variety of white pine species. But generally what we look at is there are two types of resistance that tend to exist within forest species. And they may have one or both of these, but major gene resistance, this is a pattern of inheritance that is simply inherited, right? So if you remember back to the biology classes you may have had, you talk about Mendel's patterns of inheritance. You have a dominant gene that is simply inherited and expressed fully. And in the case of a major gene resistance in a forest tree, it would provide complete immunity to a disease. The issue with that is that when we think of forest trees, they live for a long time, so they've got to maintain that immunity over a long period of time, and that is not true with the pests and pathogens, right? Their generation time is very short. They're able to evolve virulence and overcome that simply inherited resistance in a lot of cases. We've seen that in sugar pine and western white pine and now limber pine for white pine blister rust. So it's not a very durable form of resistance. And most of the breeding to this point has focused on that major gene resistance, but it's probably a waste of time and money at this point because it's just not going to be a durable solution. That's in contrast to quantitative disease resistance. So not simply inherited, hundreds, thousands of genes associated in an additive way with allowing a tree to live longer in the presence of the disease, right? It may still kill it eventually, but something allows that tree to survive for an extended period of time, even in the presence of the disease. And so we could try to pyramid these types of resistance together through breeding. We can try to enhance the polygenic quantitative resistance through breeding processes. We use a lot of different approaches. Generally, when we look at white pine blister rust, we will grow them in common gardens. We will challenge them with the disease, and then we measure the phenotypes over time using high throughput phenotyping, hyperspectral remote sensing, different greenhouse experiments, and then also using genomics to say which genes are responsible, where are they at, can we use that for marker-assisted selection or genomic selection of some sort. So a paper that kind of represents this came out this year in the New Phytologists open access Plants People Planet. And this is a project that I was pretty excited about. White bark pine has recently been listed as threatened by the Fish and Wildlife Service. And because of that, there's a white bark pine restoration plan that's going into effect. And they're trying to figure out how do we deal with all of the different threats impacting the species. And so we took 255 open pollinated families over 2 different years. We sowed them in common gardens and challenged them with blister rust, and then measured their response over a 7-year period. And what you can see, I know that the figures on here, the writing's a little bit small. The point is not to care so much about the individual lines, but it's to see this kind of spread in the distribution of the response. The top panel here, A, is the percent stem symptoms. So how many individuals ended up with cankers from the disease within a open pollinated family. And the bottom one is percent survival. The colors just represent the 2 different trials that we included in there. So the big take home here is that we had quite a huge range in response. There was no major gene resistance identified. Only 4 of the 9 white pine species have major gene resistance. But this represents what is some of the highest levels of quantitative resistance that I've seen in any of the projects that I've worked on. So much so that we see somewhere between 30 to 50% survival. That means if we take 10 open pollinated seeds, 5 of them will survive and have a decent level of quantitative resistance. That also implies that we don't actually need a breeding program to enhance that level of resistance in some cases. That it's maybe likely better just to get the seeds out onto the landscape when we identify that level of resistance so that they can survive in the presence of the disease. There is also a spatial pattern that exists where we see most of the quantitative resistance appearing kind of in the Cascades, the Siskiyous to the west, and that we had much lower levels of identified resistance on the east side of the Cascades and into the kind of Great Basin Desert in that area. So I think I'm getting close, trying to stay on track here. Let me switch then to Mitchell's project. So Mitchell is a master's student in my lab, and he is working on beech bark disease resistance. So this is clearly a problem in our area. It's been a problem for a while. You can see the map on the right here, where it was introduced kind of in the late 1800s into the New England area, and it has moved across slowly over the last 100 years. And so now it's in Michigan. It causes quite a high level of mortality, 85% usually, about 10 years post-occurrence. And the disease itself is caused by a scale insect. Scale insect gets onto the bark, damages the bark, and allows a secondary infection to get in there that will essentially girdle the tree eventually. And resistance is mediated by a reaction that kills the scale insect. We don't know a lot about the fungal component of this at this point, but only about 1% of American beech are resistant. This is in collaboration with the Northern Research Station. Dr. Jennifer Cook's lab and Dave Carey in her lab in particular have been important in terms of helping provide some of the data to us to do the sequencing on. Some of the earlier studies in beech bark disease identified patterns of heritability associated with resistance, this 1% resistance, but they weren't really able to identify markers that would allow us to see like where that resistance is, how it's, what functional traits it might be associated with. So really the first markers were identified from a GWAS here, Kalick et al., 2017. What you can see kind of above that red line is that there are 4 highly significant markers associated with major gene resistance. And you'll remember that I said major gene resistance is not terribly durable. So, you know, What that means in terms of generating resistant beech is still to be determined. But what we have now is some new genomic resources. Mitchell has access to the brand new, not even published, whole genome for American beech coming out of Jill Weisergen's lab at UConn. We've got the ability to do some different genomic analysis. And so what he is working on is a mapping family. He's going to do a linkage map from 92 progeny that were grown for beech bark disease resistance. It's a reciprocal cross. So they take the two resistant parents, breed them together, get the 92 individuals. The phenotyping that was involved done at the Northern Research Station was putting scales onto the trees, covering them up, and then after a period of time, uncover them, count the number of live scales that are still there. And you can see in the resistant example, there are no scales on there. And on the susceptible example, there are. So that's the phenotype. And Mitchell will relate the genome sequencing that he's got going on to that phenotype to try to find additional markers. First, to validate the markers that do exist, and then second, to find additional markers associated with the resistance. This is important, and I think it's also challenging because we have beech leaf disease at this point. And so you've got multiple diseases interacting. It's almost like the American chestnut story, again, where you've got these two different diseases that make it almost impossible, but we're still working on that. Okay, so Michigo tip. I mentioned earlier, Michigo tip really is a big part of my focus here at MSU. And this program started in 1974 by Jim Hanover. This is the 50th year of the program. And the goal when Hanover started the cooperative was to work with the state DNR and other industry partners to identify priorities and basically breed trees, improve trees that would be of benefit to people in Michigan and more broadly. And two major products from this effort have been genetically improved jack pine and red pine. And this is Shane over here. So his picture is not on here, but Paul Blasey has been working as kind of the tree improvement specialist for 40 years. So of the 50 years of Michigo tip, Paul has been there for 40 years, really making this happen. He has just retired here like in the last month. His retirement party is next week. So please sign up for that. He's done amazing, amazing things. And Shane is picking up where Paul left off, and we expect great things from Shane as well. But thank you for joining the team. Just real quick, Jonathan Wright, 1957, joins the department, one of the very earliest forest geneticists in the country. He's measuring trees with a bow tie on. I don't know if that's staged or if he really wore a bow tie when he did field work, but James Hanover comes in 1966, starts the co-op in 1974. Most of this is focused on provenance trials and progeny tests. Hanover does start looking at biomass production, things like black locust and some others, develops the Spartan spruce through hybridization experiments, so does some really interesting work. But following Hanover's untimely death, Daniel Keithley takes over in 1992, looks at Christmas tree species, continues some of the biofuel work, as well as pulp and timber production. Paul, of course, there through most of that. He was hired as Hanover's technician. We have a lot of genetic resources available. There have been over 500 genetic test plantings. I actually looked last night. It's like 580 genetic test plantings, 40 species or more, 15,000 a session. So there's a lot of genetic resources available within the cooperative. But we're sort of shifting gears. We're still continuing the red pine and jack pine tree improvement programs. Those are kind of the bread and butter for the Department of Natural Resources here. But through conversations with the DNR, what they're really concerned about right now is how forests respond to climate change. And so over the last 2 years, we've put together an assisted migration project to try to assess how much, how forests in Michigan will respond to climate change. And so we are putting in 12 different common gardens across the state that span 3 different soil types. So there's 4 climate environments, 3 soil types, mesic, dry mesic, and xeric. And we have 12 kind of core species, and then a 13th species, which is American chestnut, that won't be planted in all the gardens. But So we're going to test their response, measure their phenotypes across those gardens, survival, growth. We're also going to sequence them and try to use some genomic offset modeling to predict how the adaptive traits vary across the landscape. Disease resistance and durability trials with the American chestnut. And all of that, of course, is to inform management interventions in this case. And so ultimately, these trials will turn into seed orchards, and we will be developing seed that we can be used in assisted migration and reforestation projects. So these are the kind of core locations of the climate areas where the gardens are going in. We have, at this point, I think all but one of the gardens prepared in the Lower Peninsula, and we have the projects out for bid for mastication and site prep in the Upper Peninsula. That should happen this next year. So again, I just want to stress the importance of Michigotip and its role in the future of my program and where we're going with it. Ultimately, my appointment, I have 25% extension, which facilitates this directorship of the Michigan Cooperative Tree Improvement Program. But my goal is, as we get this really ramped up, is to expand membership, to really develop a series of webinars and stakeholder engagement meetings, apply the research that we do within Michigotip in my lab to inform policy and other interventions, do monitoring, and then just work with the state to really focus on their priorities. So I have one last slide. I know I went a little long. I'd like to have a little time for questions, but I've been teaching a new course, Tree Biology, Forest 175, and it's focused on really providing a background for undergraduates as they come into the program in forest biology, as they go into courses like vegetation, dendrology, forest ecology, those types of courses, that they really understand the basic biology that is driving the patterns of growth and differences in phenotypes and structure. It's a lab-based course, and so we do experiments using hybrid poplar. The students get to develop their own experiments in groups, implement those experiments, and really apply some of the understanding of biology to that experiment so they can see how plants might respond. We have clones, so they can control for genotype, they can change the environment, and then measure. And it just gives them a lot of hands-on experience that I think is going to be very valuable in the future. So, okay, that is my talk, and I'd like to thank pretty much everybody here. You know, I was listening to Asia's talk last week, and you know, she has it right. I mean, I couldn't be happier in any other department. I mean, everybody's very supportive, and so I really think that it's a fabulous place to call home. And thank you all.
Hey, good questions. So a lot of interesting things. And one thing I'm really interested in, what is, from what you understand, maybe it's not well understood, with the beach bark disease, what is it a physical mechanism? Is it a chemical mechanism?
So I think that's hard to answer, because the genome wasn't available or hasn't been available. So when we look at it, we can find outlier loci. And there's 4 very significant loci that are linked to the resistance, but there's no kind of functional annotation of that. In this case, not yet. Now, Mitchell has this whole genome sequence. The question still has more to do with how well it's going to be annotated. There's going to be a lot of work that has to go into the functional aspect of that. I think with other major genes, we see associations with like leucine-rich repeats and these different kind of proteins that might be linked to general stress response. And so I don't really know on beach bark disease what it is. It's killing the scale more than it's interacting with the fungus. So I think there's a lot to learn about that, and I don't really know at this point what it is.
Hi, I'm Bob Westfall.
Yeah. Bob, did you?
Yes. Yeah, I was a student of Jim Hanover's. I got a PhD from him in 1972 in plant physiology and genetics. I have, over the years, I've worked with Bro Kinloch on disease resistance in sugar pine. And we've not published these results yet, but we found a form of resistance which Bro calls partial resistance, where where the infected tree walls off the fungus and eventually kills it. Although in some cases the fungus remains alive, but it doesn't infect further into the stem. We found in about a generation and a half of breeding in sugar pine, dramatic increases in resistance. I wonder if you found in any indications of resistance like that in the work that you've done.
Yeah. So it was hard to hear, but what the question sounded like to me was that in several of the five needle pines, there's patterns of this term, partial resistance or quantitative resistance or polygenic resistance, in some ways are used interchangeably. And there are 9 species of white pine. They respond in different ways. So from a partial genetic standpoint, some of the work that I've done on southwestern white pine, you find exactly what you were talking about. There is a necrotic lesion that occurs around the canker. It creates a bark reaction. Crenarchum ribicola that causes white pine blister rust is an obligate biotrophic pathogen. So it needs living tissue to survive. And as that fungus gets into the bole of the tree, sometimes you have this reaction that walls off the canker. That means that the fungus can't spread outside of it. And that effectively stops that spread and provides a form of resistance. I've seen it over years where it'll wall off, you get this bark reaction, and then after some period of time, the fungus can kind of escape that. It continues to grow again in some cases, or maybe it's a new infection, but it does slow down that overall spread. I've seen that in southwestern white pine, sugar pine, western, some limber pine. The white bark pine, we didn't notice much of that in terms of bark reactions, but I have shown in a different paper on southwestern that is a major form of resistance that's related to that quantitative or partial resistance. So yes, Bob, I think that's true. We do see very similar types of phenotypes as we do with the sugar pine. And I think it's really one of the strongest types of resistance that we can use in breeding when we see something like that, as opposed again to using major gene resistance.
Correct.
Where's it going?
Thank you.
Presentation. I have questions. I don't know whether it's relevant or not, but have you done or have you seen any studies who are focusing on chemical composition of the tree correlations with the being resistance? For example, the ones have more lignins or extractive, they're more resistant.
Yeah, there are those. Oh yes. So Mojigan asked if there were some studies looking at the chemical composition of the trees as it relates to patterns of resistance, thinking about lignin production or other chemicals that might be involved. And people do look at that. I know, for instance, there are phenotyping approaches that use chemical fingerprinting to identify patterns of resistance. And I don't know off the top of my head what exactly it is doing in the chemical fingerprinting. They use a Raman spectroscopy to do this. Anna Conrad with the Forest Service has done some of that work. There's others involved. But I think that it is an avenue that could be explored. Yeah.
Okay.